Phylum Mollusca is a pretty interesting and diverse group. We have animals that are simple and worm-like, like the selenogasters or Caudofaviata, and we have animals that have relatively large brains, well-developed eyes, complex nervous systems, and the ability to negotiate mazes and learn by watching other individuals of the same species. So that's quite a bit of diversity in one phylum. Many of the animals that we talk about in this phylum you're probably already familiar with. A handful you're not, and we'll talk a little bit about both. But as far as I know, mollusks aren't like a dominant part of any terrestrial or freshwater or marine ecosystem, but they're a successful group in terms of diversity and numbers. If we're looking at just numbers, they're twice as speciose as the vertebrates. In this course, we're going to leave out some of the molluscan groups for time's sake, but what we will focus on for anatomy and physiology, at least, are the gastropods and the bivalves. Cephalopods are pretty complex, and we're only scratching the surface when we talk about them. But of course, those commonly known classes you probably are already familiar with. The gastropods are creatures like snails and slugs. Bivalves, as you know from lab, are clams and mussels and kin. And cephalopods are octopus, squid, nautilus, cuttlefish, and the like. Some of these lesser known classes you may have heard of. So Polyplacophora, those are animals called chitons, and they're shelled mollusks that have articulating plates and they cling to rocks in the intertidal zone. Scaphopoda, if you want to learn more about them, please crack open the book and read, but monoplacophorans are are similar in some ways to the bivalves, at least superficially, except they only have one valve. Caudofaviata and selenogasters, well, we're not going to talk about selenogasters, but Caudofaviata are simple vermiform worm-like mollusks that retain some of the ancestral characteristics of the phylum. And some representatives are over to your right, A, B, and C. Now, B is one of the slug species, but what I want you to notice is that opening in the side, that's called a pneumostome. So they're in a group called pulmonates, and that hole leads to a lung-like structure in the mantle cavity. Below that are sea slugs, and well, up above that is a bizarre creature that you're probably not too familiar with. We'll get to it in a bit. So just remember that these are triploblastic coelomate protostomes that are lophotrochozoans. Okay? Again, triploblastic, 3 germ layers. Coelomates, they have a body cavity, although the body cavity is reduced in some forms. Protostomes, which means they retain that pattern of embryonic development that we discussed in class. And lophotrochozoans, which means, well, they either have a lophophore or a trochophore larva. In this case, they have a trochophore larva. Now, I think we did talk a little bit about the benefits of having a true body cavity. The true body cavity is complete with mesenteries. So this helps suspend organs, and there's a more complex organ system level of organization in the coelomates. So, number one, that body cavity allows room for organ development. But again, that's not mutually exclusive to mesenteries. Number two, having a body cavity allows surface area for the diffusion of gases, nutrients, and wastes into and out of organs. It provides area for storage, and what's really important is that in the mollusks that do not have a shell, the body cavity, that is Lophotrochozoans that do not have a shell and that do have a body cavity, the body cavity acts as a hydrostatic skeleton. This is similar to what we discussed before. So if an animal contracts muscles, circular or longitudinal muscles, around a fluid-filled chamber, and the animal's soft-bodied, it allows it to elongate or shorten. For instance, that's two of the movements. The body cavity here provides a vehicle for eliminating wastes and reproductive products from the body. And if you have a body cavity, you can be larger. Okay, so we'll look at some of the specifics of how the body cavity is reduced in some of the mollusks, and later when we get to the annelids, for instance, we'll see how it's greatly expanded. Mollusks have a two-part body. They have a head, foot, and a visceral mass. Some of this is already familiar to you because we looked at and explained some of these characteristics in lab. They also have a mantle, and the mantle can secrete a calcareous shell, and it covers the visceral mass. Okay, so head, foot means, well, one of the primary structures that they use for moving is in line with the head, and the visceral mass is the region of the body that contains the viscera, or the bulk of the organs. The mantle cavity is underneath the mantle, and it's functioning in excretion and gas exchange and getting rid of wastes and releasing gametes. The mantle cavity is what's acting as a lung in the pulmonates. You know this already, but mollusks are bilateral, and they're protostome Lophotrochozoans. Now, they do have a body cavity. They have a coelom. When there's a shell, that coelom becomes a little bit less important. Well, maybe that's not the right word. It becomes, they don't rely on it so much for movement. So in the bulk of the mollusks, the coelom is reduced to cavities surrounding the heart and nephridia and gonads. And when we opened the bivalve, that cavity was called the pericardial cavity. Most of the mollusks, except for the cephalopods, have open circulatory systems. The open circulatory system means that blood, or their equivalent to blood, leaves blood vessels and enters a chamber in the body and comes in direct contact with organs and tissues. Another characteristic of the mollusks is a structure called the radula. And a radula is a rasping tongue that they use to feed. Obviously, not all mollusks possess a radula, but most of them do. Here's a generalized gastropod for you. I'm just going to say a couple things about this slide. The mantle cavity, which sits below the mantle, usually houses some respiratory structure, either a gill or a sort of pseudo-lung. The mantle, if you recall, is the layer of tissue that secretes the shell, and there's different layers to the mantle, and the different layers of the mantle secrete the three different layers of the shell. We can't really see the radula here, so I'll wait till we get to another slide to describe that, but you can see the large head foot, and there are retractor muscles depicted here. I'm sure you remember what those were like in the bivalve. And everything above this big muscular foot is called the visceral mass. Okay, so head, foot, visceral mass, mantle, mantle cavity, and here's our reduced body cavity, the pericardium. When you were looking at the snails in the petri dish on the dissecting scope, you may have seen them grazing on the elodia, the vegetation that I had in there. And when they do that, they use a tongue-like structure called the radula. Excuse me. The radula is a band of tissue that sits on top of a cartilaginous bulb, and they can evert it, and they have retractor muscles that alternately contract and relax to pull this band of tissue over the cartilaginous bulb called the odontophore. When they do that, it allows these little recurved hooks to scrape up food and move it back into the mouth and esophagus. Not every mollusk has a radula. Obviously a bivalve doesn't, but many of them do, and the tooth structure on the radula is important in classifying the different mollusk groups, or species, I should say. Here is the shell of some hypothetical mollusk, a bivalve, and the mantle, right? If you recall, this is the nacreous layer, the innermost layer that touches the outer mantle epithelium, is very smooth. And again, this is if a sand grain were to get wedged, let's say right where my laser pointer is, this nacreous layer would secrete materials around the sand grain, and that's what creates pearl. Above that's the prismatic layer, and some of you probably remember me taking your bivalve and scraping away the periostracum, the outer darkened proteinaceous layer, and underneath this prismatic layer is full of calcium carbonate, and it's almost chalky in appearance when we scrape it. Okay, but if you want to know which layer does what, you can open to page 184 and find out. But basically what you need to know is that the different layers of the mantle secrete different components of the shell. The first group that we're going to talk about are the gastropods. Gastropod means stomach foot. Gastropods are animals like snails and slugs and limpets. You might not know what a limpet is. I put an image of one at the end of this presentation. Have a look. But you can find gastropods all over the place. They're out in the open ocean. They are in freshwater habitats, and they're terrestrial. If you remember, these things are also important intermediary hosts for parasitic trematodes. Let's have a look at gastropod torsion. When a gastropod is developing, the internal anatomy twists around. And basically what this allows for is certain sensory organs to be clustered around the head region of the animal, and it allows the gastropod to withdraw head first into the shell for defense. At the end of the foot is an operculum. It's proteinaceous, and it seals them up. inside of the shell. One of the detriments to this is where they excrete is positioned up near the head and gills, the anal opening. But there are modifications like grooves that carry the wastes away from gills, for instance. It's also better to have the gill positioned up in front of the head, not in front of the head, up in front of the animal, because when a snail is moving through a mucky substrate, they tend to kick up some of the silt, and this helps that turbulence that they create, that silt that follows the animal around when they're moving through the muck, that helps keep it off of the gill. Gastropods, at least most of the modern ones, are asymmetrically coiled. So the ancestral gastropods had shells that were coiled, right? But they were symmetrically coiled or conical. And they were pretty heavy and cumbersome. But as these things exited marine habitats and invaded terrestrial habitats, they needed to lighten the load. So the shell became asymmetrically coiled, and some of the organs inside became secondarily reduced and unpaired. And this helped lighten them up without necessarily sacrificing the shell's ability to protect the animal. Gastropods feed, for instance, you can see how this is very asymmetrically coiled, right? Pretty obvious. But gastropods feed by using the radula. And the radula scrapes up material off of the substrate, moves it back into the mouth and the esophagus, and there that food material gets incorporated into a mucus string. This is very similar to feeding in the bivalves. There's a mucoid mass in the digestive tract called the protostyle, and it's rotated by cilia in the stomach. The mucus string winds around the protostyle, and eventually the digestive gland, as this mucus string swells, will release digestive enzymes into the stomach and help free up material from that mucus string and break it down. Some radulas are modified. Some, for instance, will scrape up flesh of decaying animals. Some are more sort of harpoon shaped for some of the predatory forms. Now, the waste can vary. This isn't, I guess, terribly important, but some of the freshwater forms, I would imagine they're gonna excrete a very dilute waste material, whereas the terrestrial forms are gonna excrete solid pellets to conserve water. And some of that water conservation is achieved by that nephridial tissue. And if you remember, that was a dark mass of tissue underneath the heart and the pericardium. The mantle cavity is important in gas exchange. There's a gill in the mantle cavity of aquatic and marine forms. In terrestrial forms of gastropods, that mantle cavity is modified into a lung. And there's a pneumostome. Let me turn this over into the laser. Here, there's that opening here into the mantle cavity. Now, there's not a gill in here, but there's a whole bunch of vascular tissue. This has to stay moist, and gases are exchanged inside that mantle cavity. So these are referred to as pulmonates. I think that'll do it. Some of these things that are burrowers might have rolled extensions of that mantle cavity called the siphon tube, and that might help respiration while they're, not respiration, I suppose ventilation while they're in the substrate. Gastropods move, oh, and also I should mention feed largely by using cilia. So before I get to movement, let's back up to feeding. When they're feeding, what's moving the food through the digestive tract in actually most of the mollusks is cilia. But in the cephalopods, what moves food through their gut is peristalsis. That's contraction of involuntary muscle in the gut. Okay. The gastropods, most of them, have a big flattened foot on the belly, right? And we can't see it with the naked eye. We can see it with probably, well, if we have a big one, maybe a dissecting scope. But the foot has cilia all over it, and there are mucus glands. And they use cilia to propel themselves over a slime trail that's secreted by those mucus glands. For swimming, though, they could flatten out that foot, and there's these larger muscular contractions that create some wave-like movements of the lateral edges of the foot, kind of like the flatworm that we looked at swimming. Okay, but the foot can be modified for clinging or swimming, climbing, and they could navigate their entire bodies, obviously, over a variety of different substrates. So they can make it over sharp glass because of the mucus. They could make it over very rough substrate because of the mucus, right? These are soft-bodied, relatively fragile creatures, but they can navigate through some harsh habitats because of the mucus and cilia. If you want to see how some of these marine species move around, click on the sea butterfly link. I mentioned this before. Gastropods have an open circulatory system, and the hemocoel, the reduced body cavity, is the sinus that receives the blood. And their blood, like ours, transports wastes, nutrients, and gases, and it's also used as a part of that hydrostatic skeleton. So, I'm sure you've seen a snail or a slug evert its tentacles, and basically what they're doing is contracting muscles at the base of these stalks that move fluid in there to evert them, to get them to push outward. And they have specialized retractor muscles that then pull them back. So there's no extensors here. It's a hydrostatic skeleton, so to speak, that's doing the eversion. So for the purposes of brevity, we'll say that an nephridium is a kidney-like structure that allows them to get rid of waste material while simultaneously retaining water if they need it and any nutrients that may be stuck in that waste material. Okay. The nephridium opens to the mantle cavity, and then the waste leaves the animal from there. So in land snails, there's openings to the right of the mantle cavity that allows them to secrete a less toxic uric acid in a semi-solid form. It's down here. And that takes a good amount of energy to do, but it allows them to conserve water while they're doing it. Aquatic snails secrete ammonia, and that's a very, well, it's kind of toxic to them, but it's full of water and dilute, and it takes very little comparatively energy to get rid of. Okay, so typically when you see invertebrates, even vertebrates that are living on land, they tend to secrete waste material that has proportionately less water in it than aquatic, that is freshwater species. Gastropods have a suite of sensory receptors. They have a few large ganglia in the head, and they have eyes at the base or end of tentacles. And the eyes can vary in complexity from simple pits of photoreceptive cells that detect light intensity, or they can have a lens and a cornea. Okay? They have statocysts in the foot, that's their georeceptor, and they have chemoreceptors in the mantle cavity that help detect prey and predators and sediment and chemicals in the water or air. And those chemoreceptors are called osphadia. This is the link that I want you to click on. I hope it's still functional. I didn't check this one actually. Oh yes, I did. It is functional. This is an Attenborough link on slug mating. And basically, it's describing how they use pheromones and slime and copulatory organs to exchange sperm and to meet up. Okay? So when animals are, again, slow moving or even sessile and contact is kind of fortuitous, they tend to be monoecious, right? And that helps ensure that successful fertilization will take place. So when slugs, for instance, meet up, they use these copulatory organs to cross-fertilize. And of course, the term that accompanies the monoecious form is protandry, and typically that helps prevent self-fertilization. There are dioecious gastropods, but the example of gastropod reproduction that I want you to know for your short answer question on the exam is what Attenborough describes in this link. Let's move on to the bivalves. There's another phylum from chapter 10 that we did not yet discuss. I mentioned them a couple times, but these are brachiopods. Superficially, brachiopods resemble mollusks. If you want to learn more about brachiopods, have a look at page 179 in chapter 10. But what I'll say is that brachiopods have lophophores, mollusks don't, and that the pattern of symmetry between the animals is different. You can look at this image and see for yourselves, right? But in addition to the pattern of symmetry being different, get my pointer back, you'll notice that often the two valves, right, are not similarly shaped. So in this case, the upper valve is overhanging the lower valve. And the only way to get mirror images is to bisect them longitudinally. The opposite is true for the mollusks. Both the upper and the lower shells, well, I guess we shouldn't call them upper and lower, but both of the shells are symmetrical, and we get a mirror image by bisecting them horizontally. Okay? So, different pattern of symmetry. One's got a lophophore, the other doesn't. There's some other essential differences, but there are some similarities, like they both have mantles. Okay? But enough about brachiopods, and let's move on to the bivalves. This is the second largest molluscan class. There's freshwater forms. There are marine forms. I don't believe there's any terrestrial forms, but these are laterally compressed mollusks with a sheet-like mantle and shell that consists of two valves. That means bivalvia means twice leaf. They're filter feeders and ecologically important because they remove bacteria and other substances from polluted waters. There's no head and no radula. But what does that mean? If you turn to page something or other here, 204, and take a look at the phylogenetic tree here, you can see that the bivalves are structurally, we can probably agree, much simpler than an octopus or a squid. Structurally maybe a little bit more simple than a chitin, but that doesn't necessarily mean they're lower on that phylogenetic tree, right? Lower meaning closer to the ancestral group. So this may be another example of evolution just dictating environmental demands and what mutation says, and it doesn't really have a goal in mind, right? Because there is no mind, to create a sort of superior, more advanced, more complex creature. They just are, okay? So, bivalves. Sheet-like mantle, 2 broad valves, filter or suspension feeders, ecologically important. If we look at some basic internal anatomy of the bivalves, you'll note that there, of course, are 2 valves, but embryonically, these form from a single structure that then separate. There's a tongue and groove system that makes it difficult for predators to twist them apart, and these are the structures called teeth that we looked at in lab. There's a hinge ligament, and this area is configured so that when the adductor muscles relax, the hinge naturally tends to make the 2 valves spring apart. And the hinge is an area where there's more protein secreted than there is calcium carbonate. The big adductor muscles are strong. You remember snipping through those, and they help keep the 2 valves together when need be. Of course, the line of mantle attachment here, this is called the paleo line, and what's just in front of this thing is paleo muscle, and that paleo muscle is continuous with, if you remember, the aperture that allows water to move in and out of the animal. All right, so the aperture allows water to move in and over the gills. If you remember, when we looked at that edge of the gill under the dissecting scope, you were able to see somewhat of a groove. And again, these were old specimens, and that groove was not as visible as I would have liked, but nonetheless, there's a food groove. This food groove in the gill is what houses the mucus string. So in addition to the breathing, which we'll talk about in a second, the gills are used for feeding, okay? So inside the gill, not inside, I guess, if we blow up one section of the gill, you can see that there are lamellae. There are folds of the gill. Each one of those folds consists of fleshy structures that house water tubes. And the water tube allows water to be drawn up and past blood vessels. This sets up a countercurrent exchange system. And I'll explain how the countercurrent exchange system works in a little more detail in class, but for now, know that the way the water is moving in relation to the blood that's flowing in those blood vessels up here allows the blood to pull proportionately more oxygen out of the water than if they were flowing in the same direction. And that's called a countercurrent exchange system. Okay? So onward. Please click on this link. It should take you to a Yukon site, and that Yukon site should have a couple of links on bivalve feeding. The one you are to click on is Mytilus edulis. But in terms of feeding, just in general, please look at the flow cytometry of this hyperlink. But water is drawn over the gills, and eventually microorganisms, bacteria, suspended matter, get stuck in these tracts of the gill. And apparently, well, this is debatable, but some people say cilia move the food along those tracts, the food, the suspended matter, to the mucus string on the food groove of the gill. That mucus string is eventually going to end up in the mouth and being attached to this crystalline style in the style sac just posterior to the stomach. Cilia in the stomach and the style sac rotate the crystalline style, and this thing, the mucus string, will wind around it and swell. And when it swells, it hits a gastric shield, and there's that will trigger the digestive gland to secrete digestive enzymes over it. The gastric shield helps to dislodge some of that food from the mucus. And then both the mucus string and the food are broken down in there, right? But just before the mouth, there's two structures, we only see one here, called labial palps. And the labial palps help remove debris that's too big from the mucus string, and they fling that out onto the mantle, and that's called pseudofeces. Okay, so bivalves, again, are filter feeding creatures, ecologically important animals. They use a food groove, mucus string, gills, and labial palp to incorporate food and move it into the stomach and break it down in the stomach and the intestine and absorb it. Okay? What's moving the mucus containing the food through the gut tract here again is cilia. Okay. This is kind of redundant, well, because these structures sort of show up again and again across the mollusks, but there's three pairs of interconnected ganglion associated with the esophagus. It's kind of a circular massive tissue, the foot, and the adductor muscle. Okay? The margin of the mantle is the principal sense organ, and it has sensory cells scattered throughout. They have photoreceptors, tactile receptors, chemoreceptors, and the photoreceptors can be relatively simple again, or they can be more complex, like the lens in the cornea, as I mentioned before. Osphadia are the chemoreceptors that are in the mantle. Okay, and the mantle also, this whole structure can be modified in a variety of different ways. Sometimes they're modified to look virtually identical to little fish, and larger predatory fish will then come nibble on the mantle, and this would be a female bivalve with this modification, like the lampsilus mussel. and they'll fire off their larva into the face and mouth of the predatory fish that's nibbling on them. And the larvae, the immature forms, have teeth on the two valves, and they clamp down on the gills, so they're parasites for a little bit of time. They'll eventually grow, develop, and fall off. But be sure to look at the Lampsilus mussel video. They also have statocysts, right? And of course, the statocysts are associated with nervous tissue in the pedal ganglion, but the statocysts, again, are the georeceptor. Okay. In terms of reproduction, most bivalves are dioecious. Fertilization is mostly external. A few are monoecious. Some are protandric. They have a larval form called the veliger larva. Okay, and that larval form might emerge from the female, right? The eggs and the young are brooded inside the female's mantle cavity for some time before they're released. But if a female is filter feeding, she'll bring in sperm. The sperm, through a series of complex processes, will unite with the egg. We might get a trochophore larva and then a veliger, right? A modified veliger is pictured over here, and this is the Glochidia larva. In the Glochidia larva, these are parasitic, and these hooks clamp down on fish gills, and the mom bivalve, again, will fire these out into the gills of the fish that's nibbling on her mantle. Let's look at that next. We saw this. We saw a crepidula larval phase in lab, right? And this structure is the velum. It has cilia, presumably for locomotion on this. Here's a little mouth situated in between the foot, okay? And we were able to see the velum and the foot only in lab. Okay, this is a modified mantle, okay? And it's modified to look very fish-like. It even wiggles like a fish, and larger predatory fish will come nibble on this thing, and then this Lampsilus mussel will, again, push the Glochidia larva into their faces and onto their gills. Watch this. Bivalves live in nearly all aquatic habitats. They can partially bury into sand and mud. They can attach to solid substrates or bore directly into submerged wood, coral, or limestone via mechanical abrasion, or they can use the valves to scrape a hole or a little depression in the substrate. Okay. Over to your left is a geoduck. That's the largest burrowing bivalve. It can reach 4 kilograms. It's a pretty heavy thing. I've seen these over on the West Coast, but this is in-current and ex-current aperture right here. Right, this is just an extension of the mantle, really. Okay. Down below, you have the blue mussel, Mytilus edulis, and these attach to substrates with proteinaceous structures called byssal threads. Over here, this is one of the giant clams in the genus Tridacna, and these things can be pretty big, maybe about a meter tall. That's pretty large, but you can see that its mantle is brightly colored, right? And they have, like the corals, photosynthetic zooxanthellae in their tissues. and that's a symbiotic, mutualistic relationship there. Over here is a giant rock scallop, and they can swim by opening and closing the valves of the shell. It's pretty funny looking. And as adults, though, they're mostly sessile. They attach to a hard substrate via secretions of the mantle, like a biological glue. So byssal threads here. We'll say it's probably a structure called cementum here. Some species are more effective borers and could create sizable divots in really hard substrates and remain relatively well protected. Okay, what else do I want to say here? We can see quite a few, we can see a handful of different phyla in these images. Can you pick them out? Okay, on to the cephalopods. Cephalopoda means head foot, and these are the most structurally complex mollusks. This lecture doesn't do them any justice. There's plenty on the web about them. There's plenty of video on their camouflaging. I'll talk about it a little bit, but you'll notice again that the cephalopods, even though they're structurally complex, they have well-developed muscular systems, well-developed nervous systems, relatively big brains, complex camera eyes, but they are not the latest group to evolve according to figure 1125 on 204. Okay, based on this image, you can see that it's pretty obvious why these things are called cephalopods. The tentacles are directly in line with their head. The cephalopods have a closed circulatory system. That means the blood is confined to all the blood vessels, and that closed circulatory system allows more efficient filtration of the blood. So yeah, we'll leave it at that. Their nephridia are closely associated with those blood vessels, and the arrangement of the blood vessels along with the positioning of the nephridia allow the blood to be effectively filtered. They have a big mantle cavity that's attached to that siphon, and surrounding the mantle cavity are circular muscles and another set of muscles called radial muscles, and they can use those to compress the mantle cavity, and it pushes water out of the siphon so they can quickly dart backward. Most of these don't have a large cumbersome shell like some of the other mollusks. That makes them more mobile, it makes them more agile, it allows them to be able to fit into little crevices of various different shapes. If there is a shell present, it's often internal and called a pen or a cuttle bone, and the external shell is present in the chambered nautilus, and they use that shell to help regulate buoyancy, and I'll describe how in a second. So the nautilus, it's called the chambered nautilus, and inside the shell are different chambers. And what you can kind of see here is there's a remnants of straw-like arrangement of tissue that moves through the chambers of the chambered nautilus. This straw-like piece of tissue is involved with regulating how much gas and fluid is in each one of these chambers. So it helps to regulate buoyancy. So when they're up top in the water column, there's more gas in the chambers, and when they're sinking, the gas is being moved out of the chambers, probably into the blood, and I would assume out the gills or tissue in the mantle cavity. I already described what a cuttle bone and a pen are, so I'm going to move forward. Cephalopods are predatory. They have well-developed eyes. If it were a longer course, we would talk about exactly how those eyes work, but we'll say they're well-developed, complex camera eyes, and they're structurally, not all of them, but many of them are structurally similar to ours, and in fact, I hate to use the word better, well, but they don't have a blind spot in their eye, so I would say that's better. We have a blind spot where the optic nerve enters the eye. There's no rods and cones there, so we can't perceive images in one spot of each eye. They don't have that problem. They use tentacles with cups and proteinaceous hooks to grab onto prey. They have a jaw and a radula, and sometimes venom in the salivary glands that helps paralyze prey. Okay? The jaw is not actually a jaw like ours, it's a chitinized beak, and they can pierce the shell of arthropods with this thing, and the flesh of fish or soft-bodied invertebrates. You'll notice, though, that one of these is modified, right? It looks very different. I guess two of them. It looks different than the other tentacles. So not all of these are used in feeding, right? They're used in navigating, they're used in mimicking other animals, but we'll see what these structures are in a bit. This is called a hectocotylus. Make sure you look at all of these links. What I'll say is these animals are master concealers. They're capable of changing their color of the skin. They're capable of changing the pattern and texture of the skin. And they do this all with a sophisticated nervous system. So you know that other animals are capable of changing colors, right? But the chameleons, for instance, are nowhere near as adept at it as they are. So a chameleon changes color by releasing hormones. They're naturally cryptic, but when they're threatened, they dump a hormone into their system and they get pigmentation changes in the skin. So the colors might appear to be a little more high contrasting. They might position themselves so they look a little bigger, but they can't match themselves intentionally to the surrounding environment. The only animals that can do that are the cephalopods. They do this through structures called chromatophores. And chromatophores are cells that are full of pigment. They have millions of different types. But these pigment sacs are surrounded by muscles. And when the muscles contract and relax, they move the position of the pigment sac in the skin so they can express whatever color they want to. color changes are used in courtship. They're used in defense. And as I mentioned, they can also match the texture of the substrates they're on, presumably through these muscles or similar muscles. The big point here is all of that is under control of the nervous system. They have a large brain, and the area related to sort of muscular dexterity is well developed. They can make decisions. They have memory. They can learn, and their ability to learn is on par with some of the vertebrates. So they can recognize shapes. They know where food is based on visual cues. One of my friends was actually, you'll meet him soon enough, working in a lab, and one of their octopuses would escape its tank at night, crawl across the floor, hop in another tank with fish in it, eat the fish, exit the tank, and go back into its own tank. They're pretty smart. So make sure you look at these links here. As part of defense too, they can ink, right? And inking is, in a lot of cases, accompanied by color changes. So an ink is a pigment that they can release in the water. So a big brown cloud. If a predator is coming near them and detects them, they might release this big brown cloud. Right before they release the brown cloud, they'll turn like the cephalopod will turn like a bright color. And then it will release the cloud and match the color of the cloud as it's darting away. Cephalopods are dioecious. This is the male's hectocotylus. At the end of this thing, there's a fleshy, spatula-shaped extension, and he uses that to move a spermatophore into the female's mantle cavity. The spermatophore is a sperm packet, and what happens is, I think after this spermatophore is exposed to seawater for a certain amount of time, it will kind of explode, and the gametes will be released, and they're released to her oviducts. The eggs are going to be fertilized as they're laid, and they can be deposited singly or in string-like masses, and sometimes they put them, they adhere them to the ceilings of underwater caves, and the females will tend these eggs. She'll protect them, aerate them, keep them clean of debris, but generally there's no real parental care, and development is direct, so they hatch out as miniature adults. This video clip is pretty funny, but it shows some of this and quite a bit of anthropomorphism. Have a look and see if you can pick up on it. As I mentioned, these things are smart animals. They can negotiate mazes, they can distinguish shapes, sizes, and patterns of objects. They can be trained to feed on things they normally don't feed on. They could avoid predators by posturing. They could mimic like the shape and color of a sea snake, for instance. They can cling to seaweed and essentially become that seaweed. They're impressive creatures. Have a look at this link here. Okay, they can remember what they've learned for up to about 4 months, and of course I mentioned this before, but they're, as far as I know, the only invertebrate that's capable of observational learning. So they can watch members of the same species execute a particular task, and then they can do it. And I mentioned this before, but typically animals that can do this are long-lived. They are not. All right. Polyplacophorans are the chitons. Polyplacophora means many plate bearer. They have a reduced head, a flattened foot, and a shell that divides into 8 articulating dorsal valves. And those dorsal valves, or those 8 articulating plates, are thought to be a form that's intermediate to the ancestral form and the advanced form. I'll explain it more when we get to the Caudofaviata. But they move over substrates in a manner that's similar to the gastropods. The foot is modified though. So it's so strong that it allows them to withstand waves, for instance, pounding against a rock in the intertidal zone. They can press the entire foot against some substrate and then pull the center of it up to create a real effective suction device. So they essentially are like a fleshy suction cup covered with protective plates. They move around in the intertidal zone and they graze on algae. They use a subradular organ to detect the algae and a radula to scrape up the algae. They use the plates obviously for protection, and if they get dislodged from whatever substrate they're clung to, they can tighten up into a ball for protection. Monoplacophorans, that means one plate bearer, have an undivided arched shell, a broad flat foot, and serially repeated gills. Let me pause there. These funny looking gills repeat all across the ventral side there. And when we see repetition in structure like this, sometimes it's referred to as serial homology in other forms. But when we see this repetition in structure, it shows up in the annelids and it shows up in the arthropods. So the fact that they expressed very basic serial repetition led biologists to think originally, or hypothesize originally, that these were more closely related to the annelids, things like earthworms, than they actually are. The serial repetition is likely a result of convergent evolution. Okay, so there's another example of convergent evolution in the mollusks, and that's with the cephalopods. Okay, this is when two distantly related organisms evolve superficially similar structures. So the cephalopod eye and our eye is very similar, but we're not related at all for the purposes of this point. Okay, the same thing applies to monoplacophora and the annelids. Sure, they're both lophotrochozoans. Of course, they're both protostomes, and sure, there's some repetition in structure, but that repetition in structure is largely because environmental conditions dictated a need for that repetition in structure, not necessarily because one group came from the other. Okay, so their foot works similar to the polyplacophoran's foot, and they're relatively new to people creatures, and they were only known from fossils until some were dredged up in 1952 off the Pacific coast of Costa Rica. Oh no, animation, I forgot to get rid of it. Caudofaviata. Caudofaviata means tail depression. These are worm-like mollusks that feed on microscopic organisms called foraminiferans. They're dioecious and they have scale-like spicules. Okay? They have scale-like spicules. And the spicules are calcium carbonate based. So these are thought to be early evidence of a calcium carbonate shell secreted by a mantle later on in the mollusks. So there are some similarities in how those spicules are secreted between Caudofaviata and I think how the 8 articulating plates form in Polyplacophora. So in terms of evolution, you would picture something like Caudofaviata, then Polyplacophora, and then bivalvia. So there's no real shell, but there are spicules. There's no muscular foot and no nephridia. And it's thought, again, that these things resemble the ancestral forms. If you want to know about selenogasters, they're similar in some ways. Have a look in your textbook, but I'm not going to ask you anything about it on the exam. Okay, so in summary, we have octopus and squid and relatives in the cephalopoda. Gastropods are snails, slugs, limpets, and similar creatures, bivalves, clams, mussels, oysters. Monoplacophora, well, I don't know them as anything other than monoplacophorans. We have one plate, serially repeating gills. Scaphopoda we did not cover. Polyplacophora are the chitons. Selenogasters did not cover. And Caudofaviata, likely a form of mollusk that resembles what the ancestors look like. If you're interested in any of these additional slides that I have on here, please tab through.